morning, and I hope all of you are staying healthy and safe. I'm Dr. Mark Israel, and I'm the National Executive Director of ICRF, the Israel Cancer Research Fund. Before joining ICRF 2 years ago, I served as the Director of the Cancer Center at Dartmouth Medical School for 16 years. Beyond my clinical focus as a pediatric oncologist, I led a laboratory focused on identifying molecular targets for the development of new treatments for cancer of the brain in children. When I retired from Dartmouth, I was drawn to the Israel Cancer Research Fund, ICRF, because I believe in the indomitable spirit of Israelis and the brilliant talent of the startup nation to have a positive impact on the suffering and death that result from cancer. ICRF is a nonprofit organization now 45 years old. To date, ICRF has raised $72 million for cancer research and has awarded more than 2,500 grants to scientists in all of the major research institutions in Israel. We currently fund 72 different cancer research projects in Israel. While everyone is focusing their attention on the COVID-19 pandemic, as we should, I want to remind you that close to 10 million people around the world will die of cancer this year. Cancer remains one of the leading causes of death in developed nations. So with that in mind, we think it's more timely than ever to launch our Brilliant Mind series, which will highlight many of the gifted ICRF-supported scientists at the forefront of cancer research in Israel, including our speaker today, Professor Sachi Fainara, whose work transcends cancer and has implications for other areas of medicine, including this pandemic. And now I have the honor and pleasure to introduce our moderator, Dr. Nancy Maysles from the University of Washington and chair of the ICRF International Scientific Council. Nancy.
Our speaker. Ronit is a professor in the Department of Physiology and Pharmacology at Tel Aviv University and an ICRF professor. She was an undergraduate at the Hebrew University of Jerusalem. She did her PhD at the University of London in polymer chemistry and cancer nanomedicine. She was a postdoc with the late, great Judah Folkman at Harvard in Boston, and she returned to Tel Aviv University in 2006. She has a very long list of accomplishments. In practical terms, I can summarize it as her being an inventor on 55 patents. She's recognized by numerous prestigious grants and prizes, honorary lists, including Forbes and Globe's Woman of the Year last year. She represented Israel at the 2016 Biennale in Venice on the influence of medicine on architecture. She's on the board of directors of TIVA and on the board of governors of Tel Aviv University. She currently holds the Kurt and Herman Lyon Chair in Neurosciences and Nanotechnologies. She's head of the Cancer Research and Nanomedicine Laboratory and head of the Tel Aviv University Con 3D Printing Initiative. The ICRF website has more biographical and autobiographical information, and I encourage all of you who are interested to look there. It's especially interesting to me to learn not just what other people recognize Professor Sachi Fainara for, but what she says about herself. And on the autobiographical piece, you'll find her saying, "Be brave in your choices, embrace challenge, and chance break. Just like in science, so much of life is unknowable in advance. You have to do the experiment to get the answers." And with that, I'd like to get some answers from Ronit about some of the work that's currently going on in her lab. So first, very briefly, can you tell us how you came to be working in, it's not this field, but these many fields all at once?
Hi, thanks Nancy, thanks Mark, and I would like to take this opportunity to thank all the ICRF and everybody who is listening for supporting my work for so many years and for giving me this honor of the ICRF professorship for 7 years. How did I come to this? So it's a long story, but to make a long story short, I think it came, all started from the naive thought of a girl at the age of 18 or 20 that I saw from close how cancer patients die from, in many cases, from the side effects of chemotherapy. Having said that, I want to clarify that, of course, they would probably have died much earlier without the chemotherapy, but seeing the horrendous effects of side effects, I thought naively that, oh, there must be something that can target, like a missile, that will target only the tumor cells without affecting and damaging the normal healthy cells. So from that naive thought, I went to study pharmacology, thinking that I can design a new targeted drug. And from that, I continued to nanotechnology, where we try to target very selectively, very specifically, only the tumor site without affecting all the healthy organs. It wasn't enough, because I realized that it's nice to play with chemistry, but we need to play a little bit with biology as well to get the full picture. And this is where I moved to Boston to work with Judah Folkman on vascular biology and cancer biology. And I think only when I came back to Israel and got my position at Tel Aviv University is when I combined all these multi-disciplines to work together. And today my lab is like that, works. There are 30 scientists in the lab coming from biology, chemistry, medicine, and engineering. And together we help each other to understand and get all the different pieces of the puzzle.
So I'm really curious to learn more about the project you're conducting as an ICRF professor on 3D printing tumors. Can you tell us a little bit about that?
Sure. We always start with a biological or medical, clinical question. And in this case, I was at a conference of the Melanoma Research Alliance where they were mentioning, a clinician was talking about the fact that 90% of melanoma patients that succumb to the disease present with occult small tumors in the brain. And this made me think that this is really not trivial. It doesn't make sense. Why would they go to the brain? It makes sense that the tumor cells will go to the liver or to the lung, because these are the organs that take care of making it simple to detoxify and get rid of unnecessary things in our body. But the brain is supposed to be protected. And then I went on and saw that also lung cancer and breast cancer also send metastases to the brain. And this came up with the idea that which mechanisms those cancer cells that leave the primary site and circulate in our blood system, why do they live exactly at the place of that it is most difficult to leave the blood vessels and enter the brain that is supposed to be so protected? And what we found out is that there is an interaction between the tumor cells and the normal healthy brain cells that start occurring. And by that, when I say start occurring, it means that by chemistry, we have proteins that delivered between the tumor cells and our neurons and other cells in the brain, that they talk to each other. And in that way, they somehow like call them to come and sit there because they have a hospitable niche that they can come and arrive there. And it's very difficult to think of a model where we can imitate what really happens in the patient. So this can be a nice hypothesis, but in order to prove it and hopefully develop, if it is correct, to develop drugs that target these interactions and to block them so they cannot arrive to the brain, it required a good model. And this is how we came up with the idea of 3D printing, using 3D printing for modeling cancer. And this is what we suggested the ICRF, and they went along with me with this crazy idea. And the idea was like that: we'll take a tumor chunk from the operating suite, from the pathology. So most of it goes to the pathology for diagnosis. We took a chunk and we create from it, we take all the cells, not only the tumor cells, but also the other cells, like the cells that create the blood vessels that supply the tumor cells with oxygen and nutrients that they need in order to grow and metastasize. We have the immune cells and other cells of the supporting niche that are all together. So as opposed to having what for years we have used as models, just cancer cells on a plastic dish that is very rigid, nothing like our organs that imitates what happens in the body. It doesn't have functional blood vessels. That this is how the immune cells arrive to the tumor. So without a model that will include these immune cells, actually we would have never seen immunotherapy, the Nobel Prize in medicine from last year, coming into fruition. So, and this has really revolutionized the whole field of cancer and cancer therapy. So we wanted to have a model that includes all these different cells, not only the cancer cells, and at the same time have it printed in 3D. So if we usually use only cancer cells on 2-dimension plastic dish, and then we put a compound on it and we see that they die, and then we are very happy, we take it to clinical trial, and we are devastated to see that about 90% of compounds fail clinical trial. It means that we have a really large gap in terms of translating our discoveries on the bench to bedside. And we hope that with these models, we can really mimic better the cancer with all the microenvironment, and we can have also the immune system. So in this case, we can not only test immunotherapies from the same patient when we print it according to the MRI of that patient, but also we can develop nanovaccines as we did for melanoma, that this is one of our breakthroughs in the last few years, thanks to this.
So tell me more about the nanovaccine for melanoma that you've been developing.
Okay, so in the last 5 years, we've been developing this nanovaccine that actually, instead of targeting the tumor tissue, we're targeting the immune cells. We are targeting cells that are called dendritic cells in the immune system. And what we created is a nanoparticle that is like 170 nanometers small ball that is covered by sugar. And the sugar is targeting a receptor that is covering those immune cells. So it's a way of decoying and tripping those dendritic cells in the body, that once we inject intradermally to our skin, we inject our nanovaccines, they trick the dendritic cells because of that sugar, that they come and they swallow it. And now that they swallow this vaccine, there are peptides. Those are short sequences of big proteins, large proteins, that are present selectively in tumor cells. In this case, we use the protein that exists in melanoma cells. It is called MART1. MART is melanoma antigen. This is how pathologists identify that this is a melanoma, and then when they stain the slides from a patient. So we use this because now that the dendritic cells eat this vaccine, they start recruiting cells like T cells, another population of the immune cells, that it is in a way like giving a dog to smell a shirt of a missing person, or of anyone that the police is looking for. And instead of needing to feed those cells, they just go with the memory of that smell, meaning those peptides, they go and look for cancer cells that have this protein, that have these peptides in them. And what we managed to create is, in a way, tricking the whole system to target the immune cells. They send their soldiers to attack the tumor cells, and by that we can do two things: either prevent melanoma, but also treat it when we see that they have already metastasis in the brain, like in the models that I mentioned before.
So the vaccine brings me obviously to ask you something I'm sure everyone is wondering. Can you use the same sort of platform to make vaccines against other targets, including viruses that cause cancer, and of course now coronavirus, which is on everyone's mind?
Yes, so actually 5 years ago, thanks to the whole field, the fascinating field of immunology and its combination with cancer and oncology, many of us became immunologists, and we thought that that is a big step forward. And now about 2-3 months ago, we all started to become virologists. So we learned from 0 to 100 virology in a crash course of 2 months. And we read all the papers that came out, about 4,000 papers around us, because in a way, about from the 30 people in the lab, 15 are working in the lab and 15 are at home. So we had the chance to get a lot of us just reading and summarizing. And during this time, we thought, why don't we take the same technology that we created for the melanoma, and in the meanwhile, we already continued for breast cancer, pancreatic cancer, and glioblastoma, brain tumor, very aggressive brain tumor. And if it worked in a modular way, just by changing the peptides inside that small ball, the nanovaccine, can we change it to peptides that are present on the virus, on the SARS-CoV-2 virus? And we thought that it will be really difficult, but apparently this is a flexible platform that allowed us to do it quite easily. Now the question is it working? And this is the major hurdle here, because we can test our vaccine on healthy volunteers, our little nice mice, that we see that they are able to produce antibodies against those peptides that are pieces of for example, the spike protein. This is the protein that is sticking out from all the pictures that we see of the SARS-CoV-2, and also other proteins that are in this virus. And we see that we manage to produce antibodies that know how to identify those peptides and bind to them, neutralize them, basically. The other issue was to create, this is called the humoral immunity. So we are creating antibodies. The cellular immunity means that we are also activating those T cells that I mentioned before, that usually we thought this is important for cancer, while the antibodies are more important for the virus world, the infectious diseases world. But here we see that with the SARS-CoV-2, the story is very similar to cancer, that we actually see that cellular response, meaning that we have the T cells activated, and then they can bind directly to those peptides through a receptor that they have. Then we see this activation is equally important to destroy this virus, or at least to destroy the cells, the human cells that are infected by that virus.
So this is really interesting, and I'm sure it's given our audience a lot to think about and a lot of questions. Mark has been monitoring the questions, and I'll turn it over to him to ask you some of them.
Sure.
Ronit, thank you so much. It was really interesting. And I think one thing that is of interest to me is that I was wondering, and I have a question here from a participant in Maine, what actually constitutes the vaccine? Is it a single molecule, or is it a collection of things that you have to put together? And if so, how do you go about assembling that?
Okay, so the idea is it's a combination of compounds, because we have the nanoparticle, that ball that is coated with this sugar moiety, that this is in order to trick the immune cells to be fed, to swallow this bait. But inside we have a combination of different peptides. Those are different short sequences, it's several amino acids that constitute part of those proteins that like the spike protein that sticks out. So beside that, we always need to have some adjuvants as well. In every vaccine, a while ago, it used to be either the alum or the Freund's adjuvant and other adjuvants that increases the activation of this immune response in order to get it more active and faster. And here we see that it's even more important in the case of the SARS-CoV-2, because we see that the main target population that is at risk is those vulnerable people, either elderly population or those with comorbidities that have other obesity, diabetes, and we know of many other respiratory diseases. And we see that if we manage to have a combination of compounds inside this nanoparticle, we manage to activate specific, very selective antibodies that are called, they are high affinity antibodies. They are from the germinal center. And so we don't need a lot of them. We need few, but to be very active. And this can be a solution for this high-risk population that cannot produce a lot of antibodies in a short period.
Well, that's really interesting. And it leads well to another question that has been submitted. It brings us back to cancer a bit. Namely, there are a number of cancers that are known to be caused by viruses, probably 10 to 15% of human cancer. And they would include cervical cancer, liver cancers, some skin cancers, some blood cancers. Will one kind of vaccine work against multiple tumors, or will there have to be vaccines for each individual tumor?
It will have to be for each individual tumor, similar to each individual virus. It can be, if we are smart enough, and smart enough, I mean by using all the bioinformatic tools and algorithms that we can in order to identify areas that are conserved, meaning that between different tumors of the same tumor type. It will not be a universal vaccine for all tumors or a universal vaccine for all viruses, but similar to the HPV, for example, vaccine, the human papilloma vaccine that is against the papilloma virus, and it is effective for a large population. So it means that they managed to create something that will target a conserved region. And the same is what we're trying to do here, to take a conserved region of these proteins that do not mutate, because we know that viruses similar to cancer can mutate with time. And then if we chose a region that is mutating, then it will not be helpful and will not lead to memory in a way that we want the vaccine to work. We want it to be good, like the hepatitis B vaccine, for example, that is good for 10, 20 years, and other vaccines. So we will have to make sure that two criteria are working here. One is the conservation of area that is not mutating, and it is universal for at least the same tumor type between different patients. And the second is to be able to create antibodies that will allow this memory in a way that's similar to virus, that if we are reinfected, we are not able to be reinfected and succumb to the disease. Similarly, in cancer, we will not give the opportunity to the cancer to present as a recurrence or metastasis later.
So that's an interesting point, and one of our listeners is asking, in light of your having mentioned the HPV vaccine, which is thought to be preventative and acts in a preventative manner, is there a role, and you just alluded to this, I think, for vaccines in treatment of established cancers?
Absolutely. This is what we're aiming for, because for virus, it's clear that a vaccine will not be created within a month or two. This will take longer time until approval and to see if it's really effective for long periods of time. As for cancer, most patients, and I'm not talking about the ones that are at high risk. For example, this is relevant for prevention, BRCA mutated carriers, or others that have familial polyptosis and other mutated genes that we know in advance that they have a high chance or high probability to get cancer. Most cases, we see that people come to the physician when they already have cancer. And this is why it was really important to make sure that we create a vaccine, or maybe it's better to call it an immune modulator rather than a vaccine, because we always think of a vaccine as something prophylactic, something preventive. In this case, we see that we managed to, because of this cellular immunity that we are activating, we see that we can treat cancer and not only prevent it. And not only we were able to treat tumors of the primary side, but also lead to regression when we have a recurrence and metastasis, and then regress it back, trying to reprogram the whole cancer milieu by modulating the immune system.
Well, that's really phenomenal. We have a large number of questions here, but I want to be sure for our audience that we don't go over time. Nancy, your thoughts?
So I guess there's one other quick question that I'd like to ask Ronit. Are these nano-vaccines available yet? Because we don't want to raise any false hopes for tomorrow. We just want to show what's coming in the future.
Sure. I think in general, all our work is academic. It's in the Tel Aviv University, but similar to that, most sciences in the academia can come up with ideas, test their hypotheses, create. In this case, we created these nanovaccines and tested them until the step of looking at challenging with in vivo studies, meaning on mice that have cancer. But the next step has to be done outside of the academia. For that, we need pharmaceutical companies, we need the regulatory world, and they need to scale it up and make this in kilograms. We synthesize it at grams scale, but not kilograms, to treat many patients in a clinical trial, and also in GMP and good manufacturing procedures, meaning that it has to be sterile and other requirements by the regulatory world, like the FDA in the US or the EMA in Europe, to make sure that it is safe and not only efficacious. So this will take a few years until it comes out.
Thank you very much, Ronit. I want to thank all of our listeners, our viewers, for their many wonderful questions. I see there are over 100 of you, and I hope that you've learned as much as I have. And now I'd like to turn it over to Mark to say goodbye.
Yeah, well, in this final moment, I want to thank Professor Sachi Finaro for making time in her undoubtedly very busy day to join us today, and to Dr. Maysles for pulling together this most interesting session. Tomorrow, as many of you know, is Yom HaZikaron, a day of remembrance for Israeli soldiers and victims of terror who have died. And as the siren sounds in Israel, we cannot help but think of the suffering and death caused by the COVID epidemic. But this solemn day is immediately followed by Yom HaAtzmatzu, Independence Day, which marks now 72 years of Israel's independence. These holidays are a story of light and hope. And while supporting Israel's brilliant minds working on cancer research that will benefit all mankind, ICRF believes that it's ensuring the future of outstanding medical research in the state of Israel and also supporting the state of Israel. So thank you all for watching today's webinar. Please watch for announcements of the next in this series and consider joining us in this effort to end the pain and suffering caused by cancer by going to our website, ICRF Online, one word, ICRFonline.org, and donating generously. Many thanks to all the staff at ICRF who pulled this webinar together. Have a good week, be well, be safe, and shalom from ICRF.
Thank you. It's been a pleasure being here.
Thank you.
Thank you.